Very simple topic is weed flora estimation and survey. What's the main purpose of knowing the weeds or surveying the weeds? Simply to know the weed flora which is present in that region, whether it is grasses or sedges or broad-leaved weeds. Based on that, the farmer or an agricultural practitioner can formulate a management strategy, and it is also improves the management practices taken by the farmer. If the weeds, if there are grasses, there is some management technique. If there are sedges, there are some management technique. If there are broad leaves, there are some management technique. So if you know the estimation of what kind of weeds are present in the particular area, based on that you can formulate a weed management strategy. So how do you measure the weed density of a particular area? There are two methods. One is counting method, the other one is dry matter method. So in case of counting method, what you are going to do, you are going to take a quadrant of 0.25 into 0.25 or 0.5 into 0.5 or 1 into 1 square shaped quadrant, and about 5 to 10 random places in a field you are going to keep this quadrant, and you are going to count the number of weeds present in the quadrant. You are going to calculate total number of weeds, and you are going to segregate number of monocot weeds, number of dicot weeds, number of broadleaf weeds, or number of grasses, number of sedges, number of broadleaf weeds. Based on that, you are going to calculate the number of weeds present in that particular, or you are going to estimate number of weeds in that particular area based on counting method. The other method is dry matter method. In this method, what you are going to do, you are going to cut the weeds at the base. Here, this is a weed plant. You are cut the weeds at the base, and you are going to take all these weeds and you are going to measure the record fresh weight immediately, and afterwards you are going to calculate the dry weight after drying it in the oven, and you are going to calculate the weed density. Based on that, you are going to calculate number of weeds or the population of weeds in that particular area. Precautions. What precautions you have to follow? You have to follow. You have to take care that the name and number of views should be written immediately. Afterwards, you're going to forget it. So you have to write immediately. You have to select at random places, not in a sequence. You have to take random places. If it is a field, here, 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 at random places you are going to select, and at different places in the field. And most important is you should take at the initial stage, because of the initial stage of crop growth. If you take at a later stage of the crop is already established and there is no use of taking with density, and it is also very difficult to facilitate placement of quadrate at later stage. So you have to take the initial stage of the crop growth. So here is simple calculation with density. For example, this is the quadrant. In this quadrant, how many monocot weeds per how many dicot weeds, and you're going to calculate percentage of monocot weeds, percentage of dicot weeds, and based on that you are going to convert in basis of hectare basis. 1 hectare is equal to 10,000 meters square. For example, on this 2.5 meters square, if you found two monocot weeds in the 10,000 meters square, you are going to cross multiply it, that is about 20,000 or 2,000 monocot weeds per hectare. So in such a way that you are going to calculate the density or the weed population dynamics based on the calculation based on quadrant. The other very simple formula is intensity. Weed intensity. The formula is gross total number of weeds divided by number of weeds of a species into 100. For example, if the gross weeds in a particular place is 100, number of weeds of a particular species related to Ecanova it is 20, then intensity of that Ecanova is 20. This is divided by 5. Intensity is nearly 500%, in such a way that you are going to calculate the intensity of a particular species. This is how you are going to estimate weed flora in a particular place.